Epigenetics sounds like a buzzword, but it is one of the most revolutionary ideas in modern biology. For decades, genes were seen as the ultimate blueprint. Your DNA sequence, those A, T, C, and G letters, was thought to determine everything about you. If you had a bad gene, that was it. Game over. You were destined for heart disease, for depression, for cancer. But as scientists studied identical twins, people with the same DNA living very different lives, they discovered something shocking. Twins with identical genes could have completely different health outcomes, personalities, and disease risks. The DNA sequence was the same. The way the DNA was used was not. That difference is epigenetics. Epi means above or on top of. Epigenetics is the layer of control sitting on top of your genes, deciding which ones are active and which ones are silent. Think of your DNA as a giant piano. The keys are fixed. You can't change where they are. But epigenetics is the pianist. It decides which keys get played, how loudly, and in what sequence. The same piano can produce a lullaby or a horror soundtrack, depending on how it's played. In the same way, your genome can produce very different bodies, minds, and lives, depending on how it's epigenetically performed. So, how does this actually work inside your cells? Your DNA is wrapped around protein spools called histones, forming a structure known as chromatin. If the chromatin is tightly packed, genes in that region are silent. The cellular machinery that reads DNA can't access them. If it's loosely packed, genes are exposed and can be activated. Epigenetic marks, chemical tags like methyl groups and acetyl groups, control this packaging. DNA methylation usually silences genes. Histone acetylation usually opens them up. These chemical tags don't change the underlying DNA sequence. They change how the sequence is read. Every cell in your body, except red blood cells, contains the same DNA. But a neuron and a muscle cell look and behave completely differently. Why? Because they use different parts of the genome. In a neuron, genes related to electrical signaling are active. In a muscle cell, genes related to contraction are active. Epigenetic marks lock these patterns in place. That's how a single fertilized egg cell can develop into all the cell types in your body. Epigenetics is the orchestra conductor of development. It tells cells which identity to adopt, and then enforces that identity. For a long time, scientists believed these epigenetic patterns were mostly fixed after development. But then the data started to accumulate. Lifestyle factors, diet, exercise, stress, toxins, were shown to alter epigenetic marks. Smokers, for example, carry distinct DNA methylation patterns in certain genes. Some of these changes persist for years after quitting. People who experienced severe childhood trauma show different epigenetic marks in genes related to stress response and emotional regulation. Even meditation and psychotherapy have been associated with shifts in gene expression related to inflammation and neural plasticity. Your biography is being written into your biology. One of the most famous examples comes from the Dutch hunger winter of 1944-45. During World War II, a Nazi blockade caused a severe famine in the Netherlands. Pregnant women who lived through this period gave birth to children who, decades later, were more prone to obesity, diabetes, and cardiovascular disease. Even more disturbing, their grandchildren also showed higher risk. The famine created epigenetic changes in developing fetuses that persisted across generations. The environment didn't just affect one lifetime. It left an echo in the epigenome that rippled forward. This challenged a core assumption in classical genetics, that only DNA sequence is heritable. Epigenetic marks, it turns out, can also be passed on, at least in some cases. Sperm and egg cells undergo epigenetic reprogramming, a wiping of marks to reset the embryo. But not all marks are erased. Some survive the reset, carrying information about ancestral experiences into the next generation. This is not Lamarckian evolution in the crude sense of giraffes stretching their necks and passing longer necks to their offspring. The DNA sequence isn't changing, but the way it's used can be influenced by past environments. Evolution, it seems, has more memory than we thought. Epigenetics also helps explain why complex diseases are not controlled by single genes. Take type 2 diabetes. There are genetic risk variants, differences in DNA sequence that increase risk, but they don't determine your fate. Two people can have similar genetic risk scores and vastly different outcomes depending on lifestyle. Epigenetic changes in liver, muscle, and fat cells mediate how those genes are expressed in response to diet and activity. People who are sedentary, chronically stressed, and eating highly processed food tend to develop epigenetic patterns that favor fat storage, insulin resistance, and systemic inflammation. People who exercise, sleep well, and eat whole foods push their epigenome in a different direction. DNA loads the gun. Environment pulls, or doesn't pull, the trigger. Cancer is another striking example. For decades, the focus was on mutations, permanent changes in DNA sequence that cause cells to divide uncontrollably. But many cancers show few obvious mutations in key genes. Instead, they show massive epigenetic disruption. Tumor suppressor genes, the ones that stop uncontrolled growth, are often silenced by DNA methylation. Oncogenes, the ones that promote division, are epigenetically activated. The genome hasn't changed, the epigenome has. This has led to a new class of cancer therapies, epigenetic drugs. Some treatments deliberately strip off methyl groups or modify histones to reactivate silenced tumor suppressors. Instead of attacking DNA, they attack the software running on top of it. Psychiatry is also being reshaped by epigenetics. Studies of people with major depression, PTSD, and anxiety disorders show altered epigenetic marks in genes related to the stress hormone cortisol and neurotransmitters like serotonin. Childhood neglect and abuse can leave long-lasting epigenetic scars. These marks change how sensitive the brain and body are to future stress. They don't make mental illness inevitable, but they shift the baseline. Early intervention, social support, and therapy can, over time, shift the epigenetic landscape back toward resilience. Your nervous system is plastic. So is your epigenome. All of this sounds empowering, and it is. But there's a danger in how epigenetics is often presented in pop science, as if you can hack your genes at will or think your way out of disease. That's not how it works. Epigenetic changes are probabilistic and often subtle. Eating one salad or meditating once doesn't rewrite your genome. Patterns over months and years matter. And many epigenetic marks are laid down during critical windows, like prenatal development and early childhood, when you have no control over your environment. That means responsibility is shared. It's not just your fault if your epigenetics predispose you to illness. Society, policy, and intergenerational trauma all play roles. There's also an ethical dimension to epigenetics. If prenatal environments can shape epigenomes in lasting ways, what obligations do we have to pregnant people, to children in poverty, to communities exposed to pollution? Epigenetics reveals biological mechanisms behind social inequality. Chronic stress from discrimination, violence, or instability doesn't just make people feel bad. It embeds itself into their cells. It changes disease risk. It can even affect their descendants. Health is not purely individual, it's epigenetically social. At the same time, epigenetics opens new possibilities for precision medicine. Instead of only reading your DNA sequence, doctors can read your epigenome, your current state. Two people with the same genetic risk might need different treatments because their epigenetic marks differ. Epigenetic clocks, patterns of DNA methylation correlated with biological age, can estimate how fast you're aging. Some people in their 50s have the epigenetic age of a 30-year-old. Others in their 30s have the epigenetic age of a 50-year-old. Lifestyle interventions, diet, exercise, sleep, stress reduction, appear to slow or even partially reverse epigenetic aging in some studies. Anti-aging research is now as much about epigenetics as it is about telomeres or DNA damage. One of the most intriguing frontiers is epigenetic reprogramming. Scientists have discovered that a small set of factors can reset adult cells back to a pluripotent state, similar to embryonic stem cells. These are Yamanaka factors, named after Shinya Yamanaka. Applying them briefly, in pulses, seems to partially rejuvenate cells, resetting epigenetic markers of age without erasing identity completely. In mice, this has improved tissue regeneration and extended lifespan in some experiments. The idea is radical. Aging might be, in part, an epigenetic program gone awry. If we can reset that program safely, we might slow or reverse aging at the cellular level. But epigenetic editing is a double-edged sword. Just as CRISPR allows precise changes to DNA sequence, epigenetic editing tools can add or remove methyl groups at specific sites. In theory, this could correct harmful epigenetic patterns, reactivating a silenced tumor suppressor, calming an overactive inflammation gene, or repairing stress-damaged pathways. In practice, the epigenome is complex and interconnected. Changing one mark can ripple unexpectedly. And because epigenetic states can be heritable, editing germ cells could propagate changes to future generations. The ethics of changing not just our own biology, but the biology of our descendants, are only beginning to be discussed. Epigenetics also forces us to rethink identity. If your genome is your hardware and your epigenome is your software, then who are you? The program? The machine? Both? Neither. Your personality, preferences, and vulnerabilities are shaped by a mix of genes, epigenetic marks, and experiences. There is no fixed essence hidden in DNA, waiting to be decoded. There is only a dynamic process of interaction between code and context. You are not your genes alone. You are your genes in conversation with your environment across time. This has implications for how we talk about risk. Genetic testing companies often sell reports predicting disease risk based on DNA variants. You have a 30% higher risk of X. But that's only part of the story. Epigenetic data adds another layer. Two people with identical DNA risk can diverge dramatically depending on diet, stress, and social support. Risk is not destiny. It's a starting point. Epigenetics turns genetic fatalism into genetic responsibility. Not blame, but agency. You can't change the cards you're dealt, but you can influence how they're played. And yet, it's important not to swing too far in the opposite direction and claim you can epigenetically control everything. There are limits. Some conditions are overwhelmingly genetic. Some damage is irreversible. Some epigenetic changes are too deep or too early to fully undo. The truth lies in the tension between constraint and flexibility. DNA sets boundaries. Epigenetics explores the space within those boundaries. Freedom inside limits. The narrative of DNA is destiny is comforting in a strange way. If everything is predetermined, you're off the hook. If everything is random, you're also off the hook. Epigenetics occupies the uncomfortable middle. Your choices matter, but so does your history. Your biology is malleable, but not infinitely. Your environment shapes you, but you can also shape your environment. It's less a script and more a negotiation. Every meal, every night of sleep, every walk, every relationship, every stressor. Your cells listen. They adjust. They remember. Not in words or images, but in methyl groups and histone patterns. So what does this mean for you, practically? It means that when you choose to exercise, you're not just burning calories, you're changing gene expression in your muscles, heart, brain, and fat cells. When you manage stress through breathing, therapy, or connection, you're not just feeling better. You're altering epigenetic marks on genes related to inflammation and neural plasticity. When you create a stable, loving environment for a child, you're not just shaping their psychology, you're shaping their biology at the deepest levels. The line between mental, social, and physical health is thinner than you think. Epigenetics is not magic. It doesn't replace good public health, social justice, or medical care. But it does reveal hidden leverage points, places where small, consistent changes can have outsized biological effects over time. It tells us that biology is not a one-way street from DNA to destiny. It's a feedback loop. Your life feeds back into your cells shape your life. You are both author and text. In the end, epigenetics doesn't say anything is possible. It says more is possible than you were told. Your DNA is not a prison. It's a library. Some books are open. Some are locked. Some can be dusted off and read again. Some can be closed gently. Epigenetics is the librarian, responding to how you live. You can't rewrite the library, but you can influence which stories are told, and that might be the most important kind of freedom biology can offer.